is an electron. Right? So basically, whenever a cell generates ATP, it takes some molecule that has high energy electrons in it, and it breaks it down in order to release that electron. Then what the cell does is that it uses a different molecule, some molecule that has a pull for electrons, and it uses it in order to move the electron towards it. And so that electron just moved. We said that movement represents energy, and it's actually the energy of that moving electron that the cell uses to generate ATP. Right, so again, the key idea here is that when you're talking about ATP being made, well, it's the movement of electrons that actually provides the energy to make that ATP. And so some of the basic components required in order to do this is you need some molecule that serves as a source of electrons, right? So some molecule that has high energy electrons in it. You need something to make the electrons move, right? So you require some sort of electron acceptor that has a pull for electrons. Just some terminology here. So oftentimes you might hear electron source be referred to as the energy source, right? Because it's the movement of the electrons that provides the energy to make ATP. So electron source is sometimes also referred to as an energy source. You might know that when metabolism happens, there's often waste products that are generated. And so basically where the waste products come from are here. So you see this electron source. After it's been broken down and after the electron has been released from it, basically this serves no more purpose. So all the leftovers of this molecule after it's been broken down, those are all waste products now. Same thing here. So after this molecule has already accepted that electron, its purpose has been served. Typically it can't accept any more electrons, so now this thing is useless after it's accepted the electron. So that also is usually excreted as a waste product. Right. So again, if you were to boil ATP generation down to its essence, all it is, it's all about just electrons moving from one molecule to another. And so here we go. So again, here's the essence of ATP generation. It's all about electrons moving from one molecule to another. The energy represented by the moving electron is then used to make ATP. It turns out that different cells can use different things as electronic sources, and different cells can use different things as electron acceptors. And so what we'll do is I'll show you some examples of things that can be used as electron sources and things that can be used as electron acceptors. So first, then we'll start with something that you might be familiar with. So hopefully in a previous class in cell biology, you talked about the kinds of things that human beings can use as electron sources during their metabolic process. And even if you haven't taken a class, you might be able to guess, well, what do you need in order to stay alive and get energy every day? Well, you need to eat, right? And so the things that you can eat, like we mentioned before, include, well, we can eat organic molecules, so things like proteins and sugars and lipids. And so these are the kinds of things that we use as our electron sources. In other words, these molecules are chock full of electrons that we can just, harvest out of them. You might remember that the thing that we use to tug on the electrons, the thing that we use to make the electrons move, mostly we use oxygen, right? So oxygen is a pretty electronegative thing, so it's really good at drawing electrons towards it. You might also remember that sometimes our muscle cells, in a pinch, right, if they're being worked too hard and they're not getting enough oxygen to do the work that they're being asked to do, then basically they need to use kind of an auxiliary electron acceptor in order to get a little bit more ATP being made. And so it turns out that human cells, in a pinch, when our muscle cells are being worked too hard, can also use pyruvic acid or pyruvate as an electron acceptor. So our muscle cells have some options as far as what can be used as an electron acceptor. All right. So if you were to categorize then what's going on here as far as electron sources and acceptors, that here are the terms that would be used. So we're saying that there's some cells, for example, human cells, that can use organic molecules as an electron source. And so by definition, the term that would be used to describe that is chemoorganotroph. Right, so here's the definition of chemoorganotroph. It's a cell that uses chemical compounds that are organic as their electron source. And so I underlined this to kind of show you where the term chemoorganotroph comes from. Right? It uses a chemical compound that's organic as its electron source. If you want to describe these things, so there's some cells that can use oxygen as a final electron acceptor. So the term that's used to describe that is respiration. So respiration in general just means that it's a process, or it's when a cell uses an inorganic molecule as its electron acceptor. Or oxygen is inorganic. So therefore, if a cell uses this as its electron acceptor, then respiration would, accurately describe it. We'll see that there's different types of respiration that a cell can use. Right? So if the inorganic molecule that the cell is using as its electron acceptor is specifically oxygen, then that type of respiration is referred to as aerobic respiration. Right? So respiration is when an inorganic molecule is used as the electron acceptor. If it's specifically oxygen, then it's called aerobic respiration. We said that if you're talking about humans, kind of an alternative to oxygen that can be used as an electron acceptor is something like pyruvic acid. And pyruvate, I'm showing you the structure here to kind of emphasize the fact that pyruvate has both carbons and hydrogens in it. So by definition, pyruvate, that would make pyruvate an organic molecule. So the term used to describe a cell that's using an organic molecule as its final electron acceptor is fermentation. Right? So if a cell is using something like pyruvate as its electron acceptor, then it's said to be doing fermentation. And again, fermentation is when a cell uses an organic molecule as its electron acceptor. All right. So this is kind of the, in a nutshell, the story behind how humans can generate ATP. Now if you compare that to how prokaryotes can generate ATP, you'll see that they've got us whipped. Right? Prokaryotes can do all kinds of things that we can't as far as metabolism that generates ATP. So we'll show you that right now. Right, so what all can prokaryotes use as their electron source? Well, there's a lot of prokaryotes that, like us, they can basically eat proteins and sugars and lipids in order to get the energy out of them. But unlike us, there are some prokaryotes that can eat inorganic molecules too. Right? So there's some that can use inorganic molecules as their electron source. And if that doesn't amaze you, just that term, that phrase right there, how about if you consider this? If you consider something like a cigarette, cigarettes are pretty bad for you. And some of the reasons why they're bad for you are outlined in this picture. Right? It's because cigarettes contain all kinds of nasty, inorganic things. So, for example, cigarettes contain toluene. It says here it's used to clean things that are really grody. Right? It's an industrial solvent. Typically, toluene, as indicated by the skull and crossbones, is a really dangerous thing to us, to human beings. Turns out, though, that there's actually some prokaryotes out there that can metabolize toluene, that can use this as an electron source. Same with ammonia. That's pretty nasty stuff. It's used to clean toilets, right? But there's actually some prokaryotes that can metabolize it and use it as an electron source. Even things like arsenic, which are just straight up poison to us, right? It's an inorganic thing that there's some prokaryotes that can actually metabolize it, that can eat it for breakfast, use it as an electron source. So prokaryotes, when you're talking about electron sources, they can use all kinds of things that we can't. Now, if you consider what prokaryotes can use as their electron acceptor, again, they're pretty varied there as well. So the kinds of things that prokaryotes can use as electron acceptors, well, like us, there's prokaryotes that can use oxygen as their final electron acceptor. Like us, there's some prokaryotes that can use pyruvate and other derivatives of pyruvate as its final electron acceptor. But unlike us, right, prokaryotes they step it up. There's some that can use inorganic molecules other than oxygen as their final electron acceptor. So for example,